Okay, so for our first talk of this opening workshop in the SCB programme, we're going to have our four programme organisers to present 8 open problems. So it's Radek, Erban, David Holcman, Samuel, Isaacson and Kostas Zygalakis. So I think Radek's going to start.
Thank you very much. So this is the first talk, the first workshop of our program, and this talk is given by 4 people who are the program organizers. So we wrote a proposal for this program about 3 years ago, and then we had first meeting with John Tolan, who is the director of the Newton Institute, to explain us the philosophy behind Newton Institute programs. And John emphasized that we should focus on organizing a research program which will make major advances in our research field by solving some challenging open problems. Now, this is a fast evolving research field, so in order to be up to date, we decided to dedicate first few weeks of this program just in discussion what actually are these open problems in the area. So this program already started 2 weeks ago, and last week we had first 2 talks which initiated 2 working groups of the program. So our 2 Cambridge-based participants, Ulrich and Karen, discussed they are both lab-based, and they were discussing open problems which are kind of rooted in biology. Uli talk about on actin dynamics, modeling philopodia and cytoskeleton, and Karen talk about system biology of chromosome dynamics, cell nucleus, and DNA dynamics. So these are the one types of open problems based on experimental data, and they both presented some open and unpublished data and results. So we can use mathematics to explain these results. Now, this week we have first workshops, and if you look to the website, there are 2 goals of this workshop. There'll be 3 mini courses which are aimed at students, but also at all participants, just to kind of unify the language between program participants. But the rest of it will be kind of high-level overview talks, which will highlight some of the major open problems in the different areas of the program. Now, there was some cancellation for the first slot in this program, so we decided to step in and just fill it with and introduce additional 8 problems to initiate some discussion. And if some program participants still feel that some burning questions are not covered, then next week we can organize additional open problems and working groups. And by the end of this month, we hope that we'll have plenty of open problems to work on. So then from February, June, we will kind of also hope that we'll make some significant advances to address some of these questions. So I'll just introduce 2 of these problems, and I will pass on the baton to David, Sam, and Kostas to introduce the rest. So each of us will just talk about 2 questions. So the first question which I pose here is, can we simulate complex intracellular processes which take place across the whole living cell with fine atomistic resolution of molecular dynamics? So just let's first introduce why this is actually a problem which is mathematically interesting and why is it important. So let me just summarize what actually is all atom molecular dynamics. So this is the kind of smallest scales which some program participants work on. It's a method which is popular in computational chemistry, computational biochemistry, because there's a lot of information known at atomistic level and about structure and function of various molecules. But mathematically, this is just a nonlinear system of ordinary differential equations. I describe the system by a large vector, bold X, which describes the positions of all atoms in the system, and also by another vector V, which describes the velocity, as then I evolve Newton equations. So derivative of position is velocity, and derivative of velocity is just this is inverse of mass matrix, which is just a matrix which has masses of all atoms on the diagonal, and then there are forces on the right hand side, Fx. So So for purposes of this program, we are interested in biological applications, so it's about mainly simulations in water. So there are different water models to choose from, but ultimately we have to kind of evolve the systems in time. So the typical time step which we are using in molecular dynamics simulations is 1 femtosecond, which is a very, very small time step. So which means that MD molecular dynamics simulations are limited to small domains containing only several thousands of water molecules. Now, what I pose in these questions is studying processes which is happening across the whole cell, and one cell contains maybe around 10 to power 10, 10 to power 12 water molecules. So what are the methods used there? So these are stochastic spatio-temporal modeling approaches of industrial processes, and we have plenty of participants working in this area. So people who work on Brownian dynamics, for example, small dyne type simulations, and the main developer behind small dyne is one of the long-term participants, Steve Andrews, where you just don't simulate all atoms on all particles. But you just describe the important components, important biomolecular system, and describe the changes of position by the discretized Brownian motion. So you can discretize this type of models much larger time step, nanosecond, even larger, and it has been used to model intracellular processes which take place across the whole living cell. It can be coupled with coarser models. We've written in partial differential equation, reaction diffusion master equation, and we have two mini courses on these approaches later during the week. And so it's also connected with some open problems presented by Kostas and Sam. So okay, so on the face of it, this looks like an impossible problem to simulate actually in all atom molecular dynamics simulation across the whole cell, but there are plenty of mathematical questions which we could study here. So one of them is just to look maybe more simplified coarse-grained models of molecular dynamics. So we can introduce more simple models that basically you have a large particle which is immersed in a heat bath with small particles. So we can run beautiful mathematical theorems which one can prove that in suitable limits, if you have many of these small particles which are hitting the large particle, what the large particle is doing, just doing Brownian motion. So I can exploit these theorems and design multi-scale approaches which use molecular dynamics simulation in a very small part of the domain, in the parts of the domain when the molecular dynamics information is known and important, and the rest of the simulation you just simulate some coarser approaches like Brownian dynamics. So on the face of it, this is a very simple idea, but there's a lot of challenging to actually implement this idea, partially because that this equation, this Brownian dynamics, is obviously not valid on this time scale. If you go to 1 femtosecond time scale, if you evolve the system, then the force which on the particle atom which is exerted now is very similar to the force which is exerted 1 femtosecond later. So there are a lot of correlations of forces, while this approach kind of assumes that we just have a random displacement of molecules. So if you assume Langevin dynamics, you assume that there are random forces which take place which are not correlated in subsequent time steps. So just to mention another connection with stochastic dynamical systems, which we could exploit here, since this is kind of one of my recent paper which was just accepted a few days ago, is that we could start with all atom molecular dynamics and design various intermediate stochastic models which are kind of varied on a shorter time scales and which could be used on overlap regions between all atom molecular dynamics and Brownian dynamics. So I can focus on proving some theorems how to go from molecular dynamics to some intermediate stochastic models which describes not just position but also velocity, acceleration, some also auxiliary variables which can help to match this model with molecular dynamics. But also one can prove that this intermediate model in suitable limits converges to Brownian motion. So one can design multi-scale approaches which design all atom molecular dynamics in parts of the computational domain and less detailed Brownian dynamic simulations in the remainder of the domain, which could ultimately kind of partially address these big questions in this area. Just to summarize, the mathematical questions are just analysis of limits in which molecular dynamics converges to some stochastic model. And we can do it either by proving theorems for simplified heat baths or by estimating stochastic models from all atom molecular dynamic simulations. Another mathematical challenge is to error analysis of these numerical algorithms which use these intermediate stochastic models and Brownian dynamics in different parts of the computational domain. So how error depends on the time step of the simulation, on the discretization. And then ultimately we could work on developing multi-scale algorithms which can incorporate this all atom molecular dynamic simulation into small dyne, which is one of the Brownian dynamic software packages several participants of the program work on. And I should also highlight that this program will have 4 workshops, and our second workshop will include a lot of experts on molecular dynamics and be discussing these multi-scale methods for stochastic dynamical systems in biology. This second workshop is taking place at the beginning of March. So this was question 1. Now just to introduce the question 2. So the question 2 is to Just mentioning here is from the other parts of the program. So this program focuses both on spatio-temporal modeling, which I just described, but also focuses on non-spatial modeling. So it's a question, can we efficiently analyze how the dynamics of stochastic chemical networks depend on their parameters? To put in the quote, stochastic bifurcation analysis. Just to explain it, this is a question connected to modeling chemical reaction networks. So we have a state of the system described by chemicals which are undergoing chemical reactions. So in classical deterministic mean-field model will be described in terms of, non-linear system of ordinary differential equations, where this port X is now a vector of concentration of all the chemical species involved. And this depends on this vector of concentration, but also on the parameters like rate constants of this chemical reaction, which is in K. Now there are well-developed methods for bifurcation analysis for studying how the system of ODs depends on its parameters. So if I just take a model, grab it from one of my papers. This is kind of like if you look just to the red lines, this is a typical like a simulation forwarded time of this equation. So I have a nonlinear system of ODs, I give some parameters, rate constants, and I simulate the system. So this is time, this is number of molecules or concentration. And you can see that I simulated one system for different parameter values, and you can see that for one set of parameters I'm getting just the system goes to the steady state, and for another set of parameter system oscillates. So I could investigate these properties by running simulations forward in time and just kind of going through the parameter space. But there are better ways how to do it for the system of ODEs. You can, for example, just look for the solution of fx case equals 0 and look for what are the steady states and do some bifurcation analysis. You can see that there is something interesting happening between this parameter K1, 12, and 13, that systems goes from going to the steady state to oscillations. Now, what I put on the top of it is just a simulation of the same system by a stochastic model. I can see that the stochastic model is oscillating here, but also for the same parameters, but it's also oscillating here. Now, what is the stochastic model? So the stochastic model in this area of non-spatial is given by Gillespie algorithm or mathematically by continuous time discrete space Markov chains, and we'll have one mini course by David Anderson in this workshop. And there's also the whole workshop 3, which is happening in April, which will be studying non-spatial models. So that can be analyzed through chemical master equation. So these methods are just for simulating the system forward in time, which I just showed. But a big question is to actually do something similar. What is developed for deterministic models, to bifurcation analysis, to just understand if I change the parameters, how the dynamics of the system changes. For example, find the regions of parameter space in this K space, where the given chemical reaction network is monostable, bistable, multistable, or identify regions of K-stable noise-induced oscillations. So I put stochastic bifurcation analysis. I put it in quote because what I was showing before was actually a stochastic system which was close to bifurcation of the corresponding mean-field model. I could see that actually mean-field model has some clear bifurcation between going from a steady state to oscillations, while the stochastic system was kind of oscillating everywhere. This is also connected with problem 6, which will be introduced by Kostas shortly. So I think I will now pass on button to David, who will be discussing next 2 open problems.
Okay, first of all, thank you very much for coming for this program dedicated to modeling, simulations, and analysis in cell biology. So before, when Radek suggested that we should formulate a few open questions, this was a hard task because we are not exactly a field that belongs to mathematics. We are in between modeling, statistical physics, computational biology. So, what is the meaning of formulating open questions that are not necessarily going to result in theorems? It can result in an algorithm. It can result in better understanding of the subject of the biology that you have been working on. So before giving these two directions, I would like just to go with you through some ideas that we have looked at. So first of all, we think that, and this is the importance of this community that that is represented here, we think that it's a time for some kind of new mathematical physics in cellular and biology, and we made the case for this. So we just published something in Physics Today about this idea that there are new questions in biology that requires new methods that, again, are not part of classical mathematics or stochastic processes, but are in between fields and things that have to be developed. Now, I would say that this is more or less like what was quantum mechanics 100 years ago. And so we have new phenomenology. We don't know what are the equation, like what is the Schrödinger equation of the biology today is not clear. And as we have seen from Radek presentation, the notion of multiscale, of bridging scales, is certainly one of the most important aspects of what we are doing. How to simulate things where you cannot simulate at a molecular level, and still you want to have things at a macroscopic level. How you do that rationally? How we can believe the simulation and the design of the simulation is correct, whatever means correct here. And I would say, as every new field, when you have young people entering into the field, it's always hard to publish. You know, we always ask ourselves, where to publish this? This belongs to mathematics, physics, computational biology. It's not clear what's the appropriate journal for this. And indeed, we have seen in the last couple of years many new journals appearing. And not only it's true for publication, but also if you ask for young people, how do you find a position in this field? 10, 15 years ago, this was very hard. Now it's a bit easier because there was an understanding that this is an important field, not only for the biological, the mathematical sciences, but also for the biological sciences. I mean, by analyzing something, we can extract features that cannot be getting from routine procedures. By fitting something in MATLAB, you have to invent something in the procedure. And also position promotion. So that's why I think it's important to gather this field, to have all of you here, to have this community, and to face together this type of questions. So the idea is how to solve questions in molecular and cellular biology using theory. Theory means not experiments. We don't use microscope. So we use simulations, data analysis, not only to show that what we are doing is correct mathematically, but also to predict, to make a prediction, to see things that are not necessarily obvious. And so for this, I would like to focus on two directions. One is how the nucleus of a cell is organized. And second is the question, I mean, neurobiology is a huge field. As you know, there are two big initiatives, one from the United States, the other from Europe, where at the top level, it was clear that billions have to be injected if we want to understand the problem of aging and neuronal degenerative disease. We are not going to solve this by integrating by parts. But being able to analyze data, to generate model, there is a hope that it will give some understanding. Like in quantum mechanics, it's clear that the success of the Schrödinger equation is not questionable. So there is here some hope that modeling and this kind of approach will do something. Okay, so how to develop and analyze the nucleus organization. So last week, for those of you who were with us, there was a first lecture. I'm not going to repeat and to summarize what has been said. But basically the idea is, can we use polymer physics, stochastic processes, for a single locus, first passage time, in very high dimensions? So I have to explain all of this. So I will explain some of this in the introduction lecture that I will give on Thursday and on Friday. So basically, can we use some kind of classical polymer physics to understand how the nucleus is organized? We will see in a second. I'll show you pictures. In general, can we develop a general theory of looping? So for example, suppose you have a polymer that can move by Brownian motion. How long it takes for the endpoints to come together? Can we calculate this analytically? Or how do you make a simulation? Because this is a rare event. So rare event means if you do a simulation, you have to wait a long time. So you are not going to spend wait for days or months for a simulation to be done. That's why here you need to have a possibility to do coarse-grained simulation or to do some analysis. And this analysis can be done in very high dimension. High dimension is the number of beads you have on your polymer. So you have 100 beads. This is the dimension 100 multiplied by the dimension of the space. So this is 300. And so what are the challenges? The challenges are, for example, how to predict DNA repair. So suppose, during radiotherapy, to try to kill cancer cells, what are the radiation good for? You irradiate the cell to create DNA breaks, and the cell decide to trigger apoptosis or not. So the question then becomes, when you create a break, how long it takes? Is it possible that the break gets repaired? So I'm not entering into the detail of repair because this is a field by itself. How long it takes to repair, and what's the probability that the repair occurs? And if you have two different breaks, maybe you do a wrong repair. That is, you have two breaks here, one part of the DNA is repaired with the other part, which is a mistake. So how do you investigate all of these questions? So DNA repair and how genes are reorganized geometrically in the nucleus. So in short, this is a eukaryotic cell. The genetic material most of the time is inside the nucleus. The nucleus, you can see the chromatin. There are two types of chromatin which can be seen here. One is where you see the white, and the other is when you see the dark. They have different folding, different properties. But yet, the artistic picture that the chromosomes are decondensed like this is erroneous. Something looks like this, and from this you try to infer some information about the organization. For example, this is the tip, the end of the chromosome, which are called the telomeres, which can be here labeled. There's a specific possibility to label them in yellow. And for yeast, you have 32 ends, 32 yellow parts. Yet, when you look at the picture, you don't see 32, you see clusters. That is, the part of the chromosome called the telomeres, they can form cluster, and there are between 3 and 5 clusters. And actually what has been found is that these telomeres can move between clusters. So they are dynamical clusters. So this has been revealed about 20 years ago, that there is such underlying organization. And to that day, it's not clear. What is the reason for this? How it happened that they are clustered? What is the lifetime of a cluster? And this leads to interesting physical questions. What is the mean first passage time of a cluster? How long it takes for a telomere to detach from that cluster to arrive to that cluster? And so this is an example of questions that are about the nucleus organization from the polymer physics, and to try to understand why you have this underlying structure and what it is good for. So another example, in that schematic figure, this is the nucleus surface. Most likely genes, the chromosomes that are inside have genes that are off, that are not expressed. However, genes that are expressed are most likely found close to a nuclear pore where things can be exchanged from the nucleus to the cytoplasm. So there is clearly a connection between the geometrical organization and function, and yet we don't understand why is that. So again, I think this is more like a direction of thinking rather than formulating for you precise questions. I mean, for those of people who are interested, we can discuss, and this is why I would like what we would like to organize for at least part of the meeting is a 1 week, 1 hour discussion, working group about trying to find out, discussing what are the important questions, how to address them, talking between experimental people and theoreticians to see what can be formulated and what can be tested experimentally, or if we have a question in coming from out of the data, how can this be modeled? Okay, now I'm going to switch gears. So this is the nucleus. This was based on polymer models, basically, and there is also actually some diffusion processes. Now I would like to go to a completely different scale, actually to different scales, and it's about the brain, the nervous system. And let's go to micrometer. Micrometer, this is the scale of a synapse that connects two neurons. So, for example, to that day, we do not understand what is memory in the brain, where memory is encoded, at which scale, how do you retrieve information. And so, in the last 40, 50 years, electrophysiologists have attracted their attention to synapses. This is the connection between two neurons. And for example, can we understand synaptic transmission based on molecular dynamics in this microdomain? And in particular, what is electricity in a domain which is not at the molecular level and not at a full scale of a neuron, where you can apply, and this has been shown for the last 40, 50 years, cable equations. When things do not have the geometry of a cable, you cannot apply cable equations. You have to look into different models, such as Poisson-Nernst-Planck, which is a set of partial differential equations that connect the Poisson, the charges, with the Fokker-Planck, which explain the dynamics of the ensemble of particles, of charges. And yet we can ask, is it a good model, good enough, to understand how the electricity propagates in such a microdomain? And the last part of the small classes that I will present, I would like to spend a couple of, maybe half of it on this question. It's not that I'm going to give you the answer. I'm going to show you what you can do with this model and what are the limitations. How to develop an analytical theory of PNP. It's a coupled PDE. This is an open question. It's very hard in general, especially when you want to address the question of how the voltage is propagating through a single dendritic spine, which is where the synapse is located. So, correct models are still to be found. For example, in Hodgkin-Huxley, if you think about it, you have ions going in and out, but they don't speak to each other. So it doesn't mean the model is incorrect. Actually, it did great for the last 40, 50 years about excitability. But when we are interested at what happened in a microdomain, we have to question again the validity of this approach. So we have to reconnect if we want to understand this at the biophysical level. So coarse-grained models are important. And here, of course, we need to find out what are the right model for the right scale and the question we would like to answer. So in the short answer, or the short opening, is that new models are needed to understand how the voltage is generated in this domain, especially now, because there are new advance in microscopy that allow people to get information about the voltage with a nanometer resolution. So this is exceptional in time. This happened in the last 2 years, 3 years. So now we can not only do the simulation, but compare the simulation with the experimental data. So the challenge is how to predict the voltage and the voltage change inside a neuron. I would say any understanding about how memory is encoded from the molecular to the cellular level would be a tremendous step in understanding of the physiology. But for this, we need to develop the model and the theory. A completely disconnected question is how neurons find a target in the brain. You know, when if you think about development, neurons are in part of the brain and they have to find a target in the other part of the brain. How this happens? So this is an entire field, and yet there are some fundamental questions about if you have a piece of a cell, how the cell sense the environment from molecules that travel. So for example, how a cell knows that the source is there and not over there. What is the beginning of this sensing? Yeah, and how to define neuronal property from synaptic dynamics. This is, again, part of this notion of multiscale. If you have an ensemble of neurons, how can we understand the behavior of the ensemble from the local behavior of the connection? So this is like a multiscale, again, type question. So I'm going to finish now. I'm not going to go over this in detail, but just to explain to you that 10 neurons together can lead to an activity that lasts 2 seconds. If you have things in the slice, you can again have some activities that last hundreds of milliseconds. So this is millions of neurons. And yet, if you have the entire brain, there are activities, so I'm going very fast, I don't have time to explain to you all of this, that represent the activity that lasts, again, hundreds of milliseconds, which resemble the activity that I've shown you for the 10, 15 neurons. So, in short, there is some multi-scale processes. What happen at a certain scale can be seen or represented at another scale. So, I will leave with this. We have a class. There will be 3 different classes for this week. I would like also to draw your attention that I will also perform a 2-hour class every week about this different subject. I would like to offer to the possible idea here, since we are opening, or we are going into a new direction for the participants that are interested. I thought about, from all of us, think about the idea of putting forward a new journal, which could be called Mathematical Models in Molecular and Cellular Biology and Their Analysis. Something that lives between the SciAm journals and the Nature methods, where we can have at the same time modeling, simulation, and data analysis. And I would like to thank those of you who have contributed to the Springer book for this session. Now I'm going to leave to Costa for the next open questions.
Yep. Hello everyone, and thank you for coming. So when Radek asked me to be part of this of this talk, basically, I was kind of struggling in terms of what I should present, in the sense that we already have had 2 excellent introductory talks. So what I'm planning to do, I only have a very few slides, and I would like to try to be a bit more concrete in a sense, talk about some new and at least in my mind exciting work that we've been doing, what we've done very recently, and it kind of ties in very nicely with some of the problems that we've already seen. So I'll try to be a bit more concrete. Actually, I on purposely try to avoid having slides with equations. So basically, I'm just going to try and explain a bit what this cartoon is all about. So one of the questions which I guess resonates a lot with things that we heard from Radek and David, and it will also resonate with things that we're going to hear from Sam, is how to simulate chemical reactions across a range of characteristic timescales. So I'm going to come into this problem from a different point of view from Radek. Radek went really from in the fine scale, and then he's talking about spatial models, and then he mentioned non-spatial models. I'm just going to talk about spatial models, and basically the characteristic thing there is you simulate your system with a Gillespie algorithm, or you model it with a chemical master equation. I mean, there are several good things about this. There are several bad things about it. So then people have proposed intermediate modeling regimes such that the chemical Lanzevine equation or the reaction rate equations. And all these models are sort of like have some pitfalls and some good things, and you can use them to sort of like model chemical reactions for different time scales. So this is recent work with Radek, Erban, and Andrew Duncan, where basically we started by thinking a numerical method for simulating chemical reactions across a range of characteristic time scales, but actually what we sort of came up at the end after thinking a bit about the problem was actually, I mean, I would call it a new type of model. It's not really new in the sense that it kind of brings together two different models, the one being the chemical master equation, the Gillespie algorithm, and the other being the chemical Lanzevine equation in the chemical Fourier-Planck. Basically, this cartoon really describes what this model is all about. I'm basically introducing what I call a blending region in which both of the models coexist. In one, on the left, if you like, of this blending region, I just simulate discrete events, so it's just a standard chemical master equation or Gillespie algorithm, if you like. For another part of the domain here, in which basically I have a lot of molecular copies, if you like, that's one way to think about it. I'm using the chemical Lanzevine equation, and then there is this tricky region in the middle called the blending region, in which somehow both of the models coexist. So that's basically the main idea, and if you like, it's one possible vision about how it would be possible to simulate chemical reactions across a range of characteristic timescales. What I sort of like about this is that we were able to do basically some mathematical analysis. There is theory, some theory if you like, which basically shows that at least what we're doing is not hopeless in the sense that it's at least as good as the chemical Lanzevine approximation for the chemical master equation in the volume of in the large volume limit. So that's the only slide with equations. And now here is the part that I'm going to talk about things that I really don't know, and that's also the other question that I'm going to talk about. In the sense that, I mean, when I was asked to be involved with the organization of this program, I was very excited because I think one of the things that is great about the Newton Institute is that a lot of different people from different countries come along with different expertise. So in a sense, I'm going to be a bit selfish and going to talk about some open problems in the sense of what I've been working on and how there are people here that know much more than me, and I would like to take this opportunity and sort of work with them if they're happy. So one of the possible things that we could do, and this is sort of like relatively easy, I think, is to try to extend the model in the same, I mean, here we have an area that blends sort of like chemical master equation and chemical Lanzevine equation. I mean, we can try, if you like, to create a complete model that takes reaction rate equations into account. So there's another blending area between the chemical Lanzevine equation and the ordinary differential equations. So I think there's still interesting mathematics to do in terms of obtaining estimates in the limit of large volume for the complete model. I mean, now it comes a bit of advertising for the talks that we have here. And once we have that model, and we would be interested to develop efficient numerical methods for the complete model. I mean, there are going to be actually loads of talk about this from many experts during this week, and some of these people will be in-house for the whole semester, like David Anderson, Des Chaim, and Mike Giles. And another thing that I think would be very interesting to see is now try to work the other way around. So I talked to you about the sort of like the non-spatial model. So one thing that we could try to think about is if we can extend these sort of ideas to apply in non-spatially homogeneous systems. And in a way, I mean, these are kind of interesting questions. I mean, I don't know if all of them actually have an answer. One of the things, or one of the obvious questions, is what would the meaning of the blending area be in this case? And actually, how would you be able to simulate dynamics within the blending area? In the non-spatial case, where you have two well-defined models, it's kind of relatively easy to think how to simulate the dynamics. And also, I mean, what would be very interesting to try and see, especially I think this is something that could be very good about gathering all these people here, is to try to incorporate the new model. I mean, not just that new model, any kind of new progress that we do in terms of multi-scale systems in existing spatial stochastic simulators. And again, we have a house expert in here that is staying for the whole amount of time. Radek already mentioned his name is Steve Andrews. So the last slide and the last open problem, which again, I mean, in a sense, it's not an open problem. There are a lot of people working on this, but it's a very challenging problem. And I think it's also a very important problem in the sense that we can sit down and sort of like do our little mathematical models and do our stochastic simulations, trying to describe systems. But it would be very good if we can sort of like use some of this knowledge to help in terms of estimating parameters for chemical systems coming from experiments. So again, I mean, there's going to be, again, there is someone that is much more expert than me on this topic. So this is Darren Wilkinson, who's going to speak on Tuesday. And by the way, after Darren's talk, we're going to have a photo taken for the workshop. So we'll just meet in the lobby before lunch. So in terms of the model that I was speaking before, I mean, he will explain, I guess, what these terms means. But in the model that I described, which is sort of like a jump diffusion model, part of the likelihood now is tractable, and maybe we can exploit that in terms of doing parameter estimation. And another interesting question is, with respect to that model, is how does it compare with other approximation approaches such as the linear noise approximation? And then, I mean, again, this is kind of something that I don't know a lot about, but I think it's quite interesting. It relates with the bifurcation analysis that Radek talked about, stochastic chemical systems. Maybe that would be a way, using this sort of hybrid model, if you like, and using sort of like tensor methods to try to do parameter estimation and stochastic bifurcation analysis. And with that, I'm gonna hand over to Sam.
Okay, thanks. So if you ask for people that all work on similar stuff to tell you what their open problems are, you're going to get a lot of overlap. Luckily, that means I can go kind of fast through my first one because a lot of it's already been said. So Radek kind of mentioned getting to whole cell modeling, and in particular, my interest is in how can we get to looking at things like signal transduction, gene regulation, or longer time processes like cell cycle, in ways that we can have kind of accurate and efficient methods to simulate these processes and make very useful or hopefully useful predictions to biologists. So kind of towards that end, some problems that relate to this that I see. One is a better understanding of the rigorous relationship between the different types of models we might look at. So in terms of my own research, I look a lot at particle-based stochastic reaction diffusion models. And there, I think there's several open questions that one could look at. For example, how you take these kind of particle-based models and can rigorously coarse grain them back to reaction diffusion PDE models. There's lots of work in different contexts about going from particle models to reaction diffusion PDEs, but there's much less in the kind of chemical kinetics context. And then, as Radek kind of mentioned, kind of the other direction that one would be very interested in is in these types of particle models, which I'm going to go into much more detail in the tutorial. How can one kind of scale down from them and calibrate them to be consistent with molecular dynamics or even quantum mechanics models? How do they arise out of those types of models? So a kind of second question related to this that at least is of interest to me is, how can we kind of scale up so that we can actually look at disease dynamics and evolution? These are much longer timescale problems than kind of simulating chemical processes, which is what I primarily do. And here we really need hierarchies of kind of these couplings where we can go all the way from the molecular or the quantum dynamics level up to kind of population and behavioral models. And of course, there's lots of work on coupling individual pieces, but getting a whole hierarchy is something that is obviously a very challenging problem, but would be very interesting to try to look at. And then, as Kostas just mentioned, a very big problem is how do we actually get experimental data into our models? And so again, for very specific experimental studies, there may be techniques for how to take that and extract model parameters out of that, but it'd be nice to have kind of more automated tools for doing this so that we don't all have to be an expert on these kind of data assimilation processes. And since I figured we're finishing here and you all want to go to lunch soon, I thought I'd show you some pretty pictures for the last question. So kind of changing gears a little bit, for me, one question that's very interesting is when we're thinking about cellular processes and how they work, what is the role of the cellular environment? How does the cellular environment come into play? So for example, one thing is, how does the kind of cellular substructure within a cell influence cellular processes? So here you see a reconstruction from X-ray CT imaging of a human B cell. In yellow, you can see the cell membrane. And in here, you can see all the organelles that we were able to segment inside the cell. So in blue, you see ER, endoplasmic reticulum. Purple, you see mitochondria. And whatever that color that I can't identify, but maybe it's pink or orange, you see the Golgi. And in yellow, you see the nucleus. And so one question is, if we try to take into account this cellular substructure, what kind of changes or predictions might it make in our mathematical models? What role, if any, does it have in the kind of dynamics of these processes? Now, a second component that Steve Andrews and I were chatting about the other day, and he was really stressing, is that very often in many models, we don't take into account there's this giant filament network throughout your cell. So here what you see are actin filaments in red, and let me just play the movie. This is all from within one cell. It's a COS7 cell, which is maybe monkey kidney. And this is live cell imaging from Eric Betzig's group at HHMI. I think it's light sheet microscopy. And what you see very clearly in the movie is that these actin filaments have very dynamic behavior. Here we're looking on time scales of, 10 minutes roughly. So we see these kind of very well-developed, very dense networks with very dynamic behavior, which we often perhaps aren't taking that into account. And here's just a second picture that maybe shows it clearer at the whole cell level. So here the purple is the actual actin. And again, we see very clearly that the networks are really dense. They're everywhere, and particularly where the cell is really doing a lot of the kind of morphology is changing. We see the networks are really forming and reforming dynamically in time. And you have to think about many cellular processes are taking place in this type of environment where you have these filaments dynamically updating and moving around and growing. And finally, one last kind of piece that I think is very important and often gets ignored, and this is, I think, a very challenging modeling problem if you're trying to study cellular processes, is how to take into account the fact that cells are not static. That is, the geometry inside the cell and of the cell itself is changing in time, and the cells are often moving around and interacting with other things. So here you're going to see a T cell in red interacting with the target cell, I don't remember what kind, forming an immune synapse. And you can see the cell really has quite a bit of motion. You have these sheets of membrane kind of moving along the surface. But it's very much a dynamic process, and any kind of immunological signaling, like T-cell receptor signaling that's getting activated, is happening in the context of this immune synapse, which is kind of dynamically forming. So these are kind of things features that I think would be nice to play around and try to understand better how to incorporate into our mathematical models. The experimental data is definitely now getting there in terms of these types of imaging studies. So the question is, can we now assimilate this into our models and use it to make useful predictions? So kind of just to summarize, we had all these questions, and since we're running out of time, I won't go through them again, but it'll be up on the web if anyone wants to look at it and think about them. And just to kind of conclude by thanking everyone for coming to the opening workshop and, of course, to the program in general. One of the goals of this workshop is to try to come up with problems for people to think about over the next 6 months as the program runs. So in your talks, please tell us about what you think are interesting open problems going on right now, especially if there's things that you're looking to collaborate or chat with other people about. Use this as an opportunity to put that forward to the rest of us. So with that, I'll conclude. Let me just say the next talk, I believe, is at 1:30 in an hour. Okay, so please try to be back then. And otherwise, have a good lunch. I'll see you all later.
I was going to say something.